Okay, so today we're going to finish up very quickly with the last couple of hormones that we were talking about as regulating solute concentrations in the plasma. We left off with the RAS system, the renin, angiotensin, aldosterone. So we'll briefly touch on A&P today. We're going to, for the purposes of the exam, we're going to push our last packet. So we're not going to try to rush through that stuff. I've set up the review a little different for today because I like to switch things up. I made you some practice quizzes that are now in the exam 3 folder. They don't cover everything. I wrote them pretty quickly, but they'll allow you to check yourself. So after we go through this stuff, we'll work on those basically. So I'm going to try to target really quickly these next couple things to what I want you to know. So when we left off, we were talking about the renin, angiotensin, and aldosterone system. So we saw that this works a lot like our clotting factors did, right? Our liver is making angiotensinogen, which is circulating around in our blood. And the way that renin works, that it gets secreted by juxtaglomerular cells in the kidney, right? They're secreting renin in response to things they're sensing about the tubular flow. So that converts angiotensinogen to angiotensin 1. We then have enzymes in the capillary endothelial cells in that wall that convert it to angiotensin 2, which ultimately tells us to release aldosterone, which is what's directly affecting our solute concentrations. And we saw that the reason we really care about this is that that last step where we secrete aldosterone, right? So when we secrete aldosterone from the adrenal cortex, this is ultimately going to have an effect on our blood pressure, on our MAP, because it is going to trigger a pathway we've talked about previously, the secretion of ADH. So it kind of moved backwards, right? So this RAS system that ends in creating angiotensin 2 and aldosterone increases sodium reabsorption in the kidneys, right? So really what we're doing here is making the plasma saltier. As we make that plasma saltier, as we osmolarity of the plasma, we remember that one of our triggers for secreting ADH was our sensors that can sense the osmolarity of that plasma, right? In the hypothalamus, we had the OVLT receptor, right? So as we increase sodium content, we're increasing the osmolarity of the extracellular fluid at those receptors, telling our hypothalamus to tell our posterior pituitary to release ADH, which tells us to reabsorb water, ultimately bringing our plasma volume back up and increasing mean arterial pressure, our blood pressure. So that's the ultimate kind of role of this whole flow. So that's what we did before. We left off on the stimuli for the renin release, right? So we were talking about what's happening here, right? Like why are we releasing the renin here? That's kind of kicking this off in a way. So the reason we're going to have renin released is basically comes down to different ways we can tell that there's falling pressure in the body. So blood coming through the systemic circuit, getting into the kidney, getting into the nephron, right, is ultimately getting into our nephron by going through the afferent arteriole, which leads us into the glomerulus, right, which is this first part of the nephron, part of our renal corpuscle. Right. So if we have decreased pressure in this afferent arteriole, that tells us that we're not getting as much blood to the kidney as before. So that, in a way, may be a signal of lowered overall blood pressure. We also have sympathetic activity to the kidneys that would tell us this more directly, potentially. And we also have parts of this juxtaglomerular complex, right? Specifically, we've seen our macula densa cells, right? Parts of this juxtaglomerular complex are able to sense the concentrations in the tubule filtrate that's about to leave the body, essentially. This is our distal tubule after we've looped through the loop of Henle, and then we're preparing to loop back down and excrete this fluid as urine. So if we see decreases in these concentrations of sodium and chloride in the distal tubule filtrate, that actually is usually a sign that they're not getting as much filtered into the nephron in the first place. And since we have obligatory movement of the sodium and chloride into the medulla to maintain its osmolarity, if these are super low, that's usually because we didn't get enough flow into the nephron. And so that's another kind of roundabout signal that we don't have enough blood coming to the kidneys, probably because we don't have enough blood flow in general. So those are stimuli for renin release. Okay. And this is just showing us this flow overall, right? How that would be the case, right? So largely a drop in mean arterial pressure ultimately is going to tell us to release renin. It's doing this either because we sense the afferent arterial pressure dropping directly, or because we have a drop in the glomerular filtration rate, which is just telling us, right, how much fluid is coming out of the plasma and into the nephron. When we have a drop in that fluid coming through, we ultimately have a drop in the concentration of our ions in the distal tubules, which tells these macula densa cells, some specialized cells, to send a paracrine signal to other juxtaglomerular cells, telling them to release renin. Okay. So the remaining things that we didn't talk about that I'm going to try and really focus you in on, like just like one or two facts about these because they are important. But I know your exam is Friday and I don't want to overload you with a bunch of new stuff. So what's going on with atrial natriuretic peptide is it's basically a signal from the heart that's kind of the opposite of what we've just been talking about. So you're always going to see it abbreviated A-N-P. Okay. And so A&P is a signal from atrial cells, so from the heart, in response to distension of the atrial wall, which really just means stretch, right? So stretch of the atria tells you that there's a lot of blood coming into the atria. It tells you that you have a bunch of pressure there, right? So A&P is going to increase glomerular filtration rate. So it's going to increase the amount of fluid moving from the glomerulus into the nephron, right? So the amount of fluid we're filtering is going to do that by dilating the afferent arteriole. So when we dilate something, right, we decrease resistance, which means we're increasing flow. So more blood is getting there. We're going to constrict the efferent arteriole. So we're basically backing that blood up, backing that plasma up in the glomerulus, which creates more time and more pressure for filtration, for movement of the fluid out of the plasma and into the nephron. Okay, so what I'm going to circle, that's our kind of like conceptual background for what A&P is and what it's doing. What we want to focus on here, I'm going to make sure we circle only like one fact, right? So let's just focus on this, right? So if you have a flashcard for A&P, let's only really know about this and that it's coming from the heart. Okay. So A&P overall is going to increase sodium excretion in response to the heart sending a signal saying there's a lot of pressure, it's really full. Okay. So increased sodium excretion. So this is the opposite of aldosterone. Like that's why we're covering A&P, because remember that the pathway for aldosterone, right, it was increasing sodium reabsorption in order to ultimately tell us to reabsorb more water. Now we're increasing sodium excretion. So we're really kind of turning off aldosterone. That's what we're doing with A&P. So if you're interested, look at all of these, but what we're just going to highlight for our purposes for today, right, is that we have atrial wall stretch releasing A&P, ultimately increasing sodium excretion and decreasing aldosterone. Okay. So this is just the opposite. A&P is the opposite of aldosterone, basically. So the final thing is potassium balance, which I kind of mentioned when we were talking about aldosterone before. So this, I just want you to know, because if you're in clinic in future, I won't just talk about sodium when it comes to talking about like dialysis and kidney problems, right? We'll also talk about potassium. So you should know that the kidneys are regulating potassium as well, right? So also crucial to function, but don't worry about memorizing these words, but know them for the future. Don't go into the future and say, I've never heard of potassium in the kidneys. Potassium is there. We are balancing potassium with the kidneys. Okay. Just like with our sodium, we can have high plasma potassium, we can have low plasma potassium, hyper and hypokalemia, respectively. This is a rundown of basically where we have already seen potassium. So we can use this basically to kind of review briefly what's going on in the different parts of the nephron, right? So in the glomerulus, everything is freely filtered, right? That's just the role of the glomerulus. The only things that don't get filtered freely at the glomerulus are we're not filtering proteins and we're not filtering red blood cells, right? So no surprise that potassium is freely filtered there. Same deal with the proximal tubules. Like their big role, the proximal tubules, is reabsorption. They do bulk reabsorption. They do most of the reabsorption. It's really not regulated. We don't see all these different hormones and signals acting on the proximal tubules. So potassium ions are reabsorbed, but it's just the same way we see reabsorption of everything else. And then the distal tubules and collecting ducts. Here we have reabsorption and secretion. And all we want to know about the potassium ions really is that they're doing the opposite of the sodium, essentially. So they are going to be regulated in the distal tubules and in the collecting duct because we're regulating the sodium. And sodium potassium pumps, right? They're two ways. So if we are regulating sodium and we are at all using a sodium potassium pump, we are also regulating potassium because that's just how that transporter works, right? So ultimately, while aldosterone is doing its job regulating sodium, it is also regulating potassium because it kind of has to. So potassium in the plasma would directly stimulate aldosterone release. And as potassium in the plasma increases, more aldosterone is released, because this is kind of just the opposite of the signal going on with sodium. So that won't appear directly on a question on your exam, but just try and kind of conceptualize that. At least remember it for the next time you hear about kidneys in a future class, right? That the potassium and sodium are kind of doing the opposite thing, and potassium is related to aldosterone. It's what we want to keep for now for our purposes for this class. Okay. So we're not going to particularly look at this, right? This is just looking at the proximal tubule. Reminder, we have pretty standard looking epithelial cells in the proximal tubule. We have lots of things that are just reabsorbed across, reabsorption being movement from the tubular fluid, right, into the paratubular fluid and ultimately into the capillaries surrounding those paratubular capillaries, which are full of plasma because we have plasma in the blood. Distal tubule, this is just focusing on the potassium part. right? We've seen the sodium part already, but this is just pointing out that as we have these sodium-potassium transporters being activated on the basolateral membrane, that while the sodium is moving this way, right? Sodium is moving this way into the blood, being reabsorbed, ultimately trying to trigger our osmoreceptors so that we'll reabsorb more water. Potassium is being secreted into the tubular fluid. So it's going the opposite direction in response to our aldosterone. And this is on the right is the picture that we saw before. We focused before on the sodium part, right? We focused on sodium, right? But we're also doing potassium at the same time. It's just the opposite direction. So sodium's going into the blood, potassium's going out of the blood. Okay. So that is all I want us to know about that. So basically, we just know that two things are opposites of stuff we talked about before. So we want to know that A&P is the opposite of aldosterone, essentially, and so it's going to have opposite effects, right? And then we want to know that whatever sodium's doing, potassium is doing the opposite, especially in response to aldosterone release. Okay. And we're not going to get in detail on these on the exam at all. If they appear, I will make sure it is super simple at that really high level, none of these details, unless we've talked about them in previous lectures, because we have occasionally seen, right, potassium in previous lectures because we've talked about sodium potassium pumps before. Okay, so I'm going to do a quick poll, and the question is going to be whether you want to talk together as a group right now to review, or whether you want to go directly into just doing these short kind of practice quizzes I've created for you. So quick poll on what we want to do now. Okay, so I am seeing it is not unanimous, and I want to make sure that you guys all get what you need. So what I'm going to do is I'm going to make sure that you guys have the link for where those practice quizzes are. So let me grab that for you real quick. They're just in the same folder as the slides, but if you don't have that up, that's okay. So I'm posting that link in the chat right now. What I'm going to do is I'm going to I am already recording this like I usually do. So I'm going to set up some breakout rooms. So if you want to go into a breakout room and just kind of quietly work through those, go ahead and do that. If you want to talk and have a kind of Q&A session, I'll stay here in the main room and do that, and you can pop back and forth, and whatever we talk about will be recorded if you decide you think you made the wrong choice and you want to come back after. So I'm gonna set it up so that you can choose your rooms. Yeah, okay. If you want to do Q&A, stay here. If you don't, just go ahead and pop into a breakout room. Since they're set up so that you can choose your own, you should be able to just kind of pop around. or I suppose you could just mute me while you're working and then unmute me after that. Okay.
I have a question.
Yeah, absolutely.
So on the practice quizzes, it does say they're for points. Is this just review or will it be graded?
It's just review. The points are just so you can kind of immediately see how you did. That's it.
Okay.
Yep. And the key should automatically pop up as well. So you don't have to do them right now in class if you don't want to.
Okay, thank you.
Yep. Okay. So who has something they want to go over?
I was just gonna ask, like, what content in terms of like the kidney anatomy we'll have to know on the exam. That was like the first chapter section we went over. I was just wanting to clarify what'll be on the exam from that.
Sure. So what I want you to know from the kidney anatomy stuff is basically what we're not covering in anatomy lab. So specifically, I'm going to try to keep it to the level of the microscopic anatomy. So let me pull up those slides and kind of indicate. There we go, anatomy stuff. Because I realize we're kind of double dipping on some of the kidney slides. Okay. So here we have the anatomy stuff. This will be quizzed in anatomy lab. So I'm going to keep that for lab purposes. These are also on your lab, so I'll let your lab instructors cover that. Okay, so it's really when we get to this stuff that I would want you to know it for the purposes of our lecture exam, because the glomerular capsule, the tubules, the nephron itself, and the cortex and the medulla are not going to be things that we will see in lab. Don't worry about most of these. What I want you to know from this slide on the blood flow, that's the kind of mix between. We'll keep it to the more microscopic anatomy parts. So the afferent arterioles, the glomerular capillaries, and then the efferent arteriole and the paratubular capillaries where they reappear later. Know those, but you can skip these larger arteries. So anything that says arterial or capillary we'll do for lecture. Anything that says arteries, potentially on your anatomy exam, although I think they're really only looking at the really big ones. Okay. Just skimming through to see if there's anything else we want to not cover. Okay, so yeah, as they appear, just continue kind of with that. So collecting duct, yes. Papilla, minor calyx, ureter, urinary bladder, urethra, that's for the anatomy lab. Does that answer your question?
Yeah, it does. Kind of just like clarifying, so we don't have to know like the anatomy of like ureters or like bladder or anything like that either then.
For most of that, no. Let's go to the bladder stuff and we'll pick real quick. So for bladder stuff, yeah, most of this will be covered in lab, so I think we don't have to worry about that. The part that is kind of unique to what we've been doing lecture is the actual discussion of how micturition works. So for the bladder stuff, the only stuff I would want to make sure that you review for the exam really is this slide. So like this stuff, because this kind of relates to how the reflex works and pressure. Know the structures on this slide, but otherwise we'll leave it for anatomy lab.
Sounds good. Thank you.
Awesome. Yeah, don't know, somehow, even with cutting down content, this exam still feels like it has plenty of things to ask questions on. Okay. So yeah, go ahead and ask any questions or general topics you'd like to go over either out loud or in the chat. Happy to address them either way.
One question I have is, are we going to, I know we're probably going to need to know the filtration, but are we going to need to know much about the different pressures?
Do know the pressures. Yeah, absolutely do know the
Okay.
pressures. Yep.
Thank you.
Mm-hmm. So since you're asking that, nobody else is asking a question right at this moment. I can talk us through those for now, but keep thinking of questions. So what she was just asking about, the pressures, right, that we're talking about, the glomerular filtration pressures. Overall, we're trying to get to this idea of an overall glomerular filtration pressure, which is going to lead to a glomerular filtration rate, although we're not calculating out the rate from the pressure. So the reason we care about these filtration pressures really is because how much flow there is through a nephron matters, right, for the renal handling of water, of these ions that we've been talking about. So the filtration pressure is really important because it's part of how that's getting controlled, and it's part of why we see all the effects that we're seeing at the end here with these hormones, even though I'm not going to ask you to go super in detail into the explanations. Like when we talk about, you know, how we're affecting the glomerular filtration rate with A&P, right, we would be kind of directly affecting this glomerular capillary hydrostatic pressure. Like that's one of the mechanisms. So even though I'm not going to ask you that directly on the exam, that's part of why for your future, if you come back to kidney stuff, which if you're going into healthcare, I imagine you will, kidney problems are common, working with the kidneys to regulate common things like high blood pressure is also really important. So ultimately, these mechanisms do come back in a lot of ways to either regulation in the distal tubules and the collecting duct, or to changing these forces for filtration. And then therefore, as we change our filtration rate, we change what gets into the kidney, which means we change things like our filtered load, which we remember is how much of a specific molecule we get into the nephron. And then as we talk about filtered load, we can talk about how close that gets to a transport maximum. So the filtration pressures are very similar also just to the forces that we talked about when we talked about net filtration pressures, when we talked about normal capillaries, right? Like the glomerulus is a system of capillaries. Now, whether you really remember what was going on with the normal capillaries and the filtration pressure there, I know that was kind of a tricky topic for some people in our previous unit as well. If you did, we can really directly relate this, right? So the hydrostatic pressure is just anywhere we have a fluid, it pushes on the walls of whatever vessel it's in. So either fluid in the glomerulus, so fluid in those capillaries, pushes out on the wall, right? And if you press out on the wall, it means you're pressing, better color, pressing into the nephron, right? You're pressing out on that capillary wall. Just extend the arrow. That means we're filtering in to the nephron. So that capillary hydrostatic pressure is a force for filtration. Versus if we go the opposite direction, right? Fluid in the nephron, pushing on this wall, right? If we extend that arrow, it's a force opposing filtration, right? So the two hydrostatic pressures oppose each other. Okay, I'm getting a question just double checking whether we're covering the acid-base slide packet. No, we're going to move that to the next exam. And before we get started on the next unit, I'll go through the whole unit and figure out exactly how to kind of scale it back and fine tune it so that we don't just get overwhelmed before our final. not trying to build up content super deeply. So I will let you know if there are big things I'm deleting from that, but that'll be after the exam. All right. So the oncotic pressures, right, our other two, we have one from the glomerulus. So glomerular capillary oncotic pressure. We have a Bowman's capsule oncotic pressure. These are our pulls. So whenever we have a hydrostatic pressure, it's a push. When we have an oncotic pressure, it's a pull. It's an oncotic pull, meaning it's the pull of like proteins dissolved in here. So we have proteins dissolved in the capillary. That's our glomerular capillary oncotic pressure, and they're pulling on things. So it's a force opposing filtration. Versus if we had a pull from Bowman's capsule, which essentially, this is pretty much zero, but the pull, the direction would be for filtration. So I'm getting a question, just making sure they got the right notes from today about potassium and sodium and A&P. So potassium is doing the opposite of sodium. A&P is essentially doing the opposite of aldosterone. So like aldosterone would tend to, I think we have the slide still open. And if we compare the actions of aldosterone and A&P, you guys see this? Oh. Okay. So like when we had aldosterone, right, our ultimate effect was that we're raising this blood pressure, right, in response to aldosterone after we go through many, many steps. So the A&P is a reaction to raises in blood pressure because that signal is coming from the heart. So it would basically do everything in reverse, although we're not going to go into detail on that. So A&P, opposite of aldosterone. Okay, I'm getting a request for the medullary osmotic gradients, which is great. That is a chunky topic, so let's pull that up. In the renal physiology packet. Still, or in the renal balance? Balance, okay. Okay, so fundamentally what we're doing when we're thinking about the medullary osmotic gradient, right, is we're noting that in our nephron, we have basically 2 points at which we're going to dip down into the medulla. We have the loop of Henle. Here we have a loop of Henle. Here we have a loop of Henle. And then we loop back around, and eventually our collecting duct for sure is going to go back through. And in a juxtamedullary nephron, which is the type we've mostly been seeing, parts of the distal convoluted tubule may also be in the medulla. So what we're doing when we're thinking about the medulla is really we're thinking about how the difference in the osmolarity of the medulla, the really increase in its osmolarity, affects water reabsorption. And really what we do, right, is we set up an increasing gradient. So we have like 300 milliosmoles in the cortex, and then in the depths of the medulla, we get up to like 1400. And the reason we do this is that as our fluid gets more and more concentrated as it's going through here, eventually going through the collecting duct, right? We have super concentrated fluid. We want to keep being able to absorb water if we have ADH present, right? But in order to do that, we would still need a gradient available, which is why we create this gradient in the medulla. So the way we do it is essentially, we have the system where we pump, let's make blue. Okay. So in the ascending part of the loop of Henle, so the thick ascending limb, we're pumping a bunch of solutes into the medulla. Other thing we have happening here actually is that we also have this solute urea joining in. And so osmolarity, right, is really just talking about the fact that we have a bunch of impermeant solutes somewhere exerting a pull on water. So when we put all these ions and we put urea into the medulla, we're basically making it salty. We're making this pull on water. And this pull on water increases as you go deeper into the medulla. This is going to have an effect on any water that has aquaporins available to it in the collecting duct. And it's going to have an effect on the water flowing through the descending limb, which is the first part of the loop of Henle. Right, so it's going to be drawn towards the salty medulla as well. So that's like our conceptual idea of what's happening here. But I think you might be asking kind of about these specific numbers, like when we went through them here. Okay, so this was just for the setup. So ultimately what we're trying to understand is this, sorry, I'm trying to be careful with my words. These differences in the concentration of our filtrate that's becoming urine as we go through this loop. So when we look at the osmolarity of the tubular fluid, when we're in the cortex, it's at equilibrium with the cortex, so it's 300 milliosmoles. As it descends into the medulla, it becomes more and more highly osmolar because it is essentially not quite reaching, more or less reaching equilibrium with the concentrations in the medulla, so with the osmolarity of the medulla. And we can note that part of the way this is happening, right, is water is moving out, right? And as we move water out of the fluid, that means that the remaining solutes are in less water, so it's going to become more and more concentrated, and that's why we have increasing numbers. But then as we move out of the medulla, as we move through this ascending limb, we are pumping our solutes. So we're taking, right, sodium, for example, also potassium, also chloride ions. We're pumping them into the medulla to keep the medulla salty. So the osmolarity of that fluid goes back down again as we go up. And actually in the distal collecting duct, we end up with a really low osmolarity as we move out of the loop of Henle ultimately here. Okay, so really the main thing we want to know about setting up this gradient is I really just want you to know what the ascending limb is doing, right? Which is that it's pumping out these ions into the medulla, which raises the concentrations of things in the medulla and therefore raises the osmolarity of the medulla. And the converse of that is that that's dropping down the osmolarity of the fluid remaining in the tubule. So that's what I want us to know about that. And then to realize what's happening on the other side, right, is that this is then going to pull on water anytime we have something move through the medulla, but specifically we think about the descending limb in the context here. Okay, I'm getting a question about the juxtaglomerular complex. There are multiple parts of the juxtaglomerular complex. So that's back in the anatomy slide. So let me pull those up for a second. Okay. So a unique part of the nephron is this region called the juxtaglomerular complex. So what we're looking at, maybe I'll zoom back out for a second, right? We're looking at this part where the nephron, right, our glomerulus, the capillaries coming through here. Our nephron goes, does its thing, it's looping around through the loop of Henle, but ultimately it comes and passes by the glomerulus again. And so if we zoom in on just that part, we have our juxtaglomerular complex. Okay, so that's here. And it has three different types of cells we're interested in. So we're interested in all three of these. So we have our macula densa cells, right? So our macula densa cells are here, right? So our macula densa cells are essentially in contact with the tubular fluid, right? Which we said that ultimately one of the reasons we're releasing renin, which is ultimately going to give us aldosterone, which is going to trigger ADH, which is going to trigger water reabsorption, is that these macula densa cells are sensing the low concentration of sodium and chloride in the fluid. So those are one component of the juxtaglomerular complex. We then have the juxtaglomerular cells themselves, which are kind of here in the middle. We also have mesangial cells, which are involved in kind of regulating GFR. So all three of these are considered part of the juxtaglomerular complex, and I'm just going to kind of show you where they come back to us later on. So one is for our regulation of GFR. I think that's here. There we go. So mesangial cells are part of one of the ways that we can regulate our glomerular filtration rate. So here, this mesangial cell contraction is happening here in this juxtaglomerular complex. So here we see our extraglomerular mesangial cells, right? So they're in here with our other juxtaglomerular cells. And they basically can help us squeeze down on the blood flow here and help us regulate our GFR. So that's one of the things that we're doing. And then the other thing we're doing with these cells is kind of what we were just talking about, right? In our stimuli for renin release, right? So here is another role of that juxtaglomerular complex in general, right? Is that we're releasing our renin in response here to cells in the juxtaglomerular complex. Move a bit back here, right? They're on this slide as well. The juxtaglomerular cells in the kidney are directly releasing renin. So these three cell types are different from each other, but they are all kind of working together ultimately. But it's also helpful to know that that one's not showing a correct answer. I will make sure that that shows a correct answer. So let me grab that. So hopefully that'll update for everybody. Okay. That is the official end of our meeting. If you have questions that come up as you're studying, feel free to email me those. I have gotten a couple emails that you would like to see your quiz responses be visible. So those weekly quizzes, you should now be able to access them all and see how you did, and also see what the correct answers should have been. So if you haven't checked those out, go ahead and give that a try. Otherwise, I don't think I have any more updates for you, but stay tuned. I don't expect to have more updates before the exam, but on the next stuff. Okay, Amanda, I see your message. So if you want to let's pop into a breakout room and see exactly what's going on with your stuff.